The Silty Pond Mussel, formerly known as the Chinese Pond Mussel, is one of the newer invasive species found in New Jersey that poses a significant threat to our native freshwater mussels and ecosystems. This mussel was first discovered in New Jersey in around 2010 when a former aquaculture and fish farm facility was purchased for land preservation. When the property was purchased, it was found that there were still several species of non-native carp present in the impoundments there. So in the process of looking to control the non-native carp, a large population of unidentified freshwater mussels was documented at the site. The state began looking into what species they were, trying to identify them, and they were found to be the Silty Pond Mussel that is native to Asia. They can grow to large size, about the size of a Nerf football, and get to high densities. Like a great many other invasive species, they're very good at what they do. They can outcompete all the native organisms in the environment. And what happens when they get to high densities like that, they actually alter the water chemistry. They remove all the nutrients in the water environment. So they outcompete the native species for space and for nutrients. The problem with this occurrence in New Jersey is that this is the only occurrence of this invasive species in the entire North America. And we want to keep it contained to where it is and hopefully eradicate it. So we're doing surveys throughout the Wickeecheokee drainage that was documented in it, as well as any other waterways that may be impacted, such as the DNR Canal, the Delaware River, or the Raritan River, to try and document if the species is in other places. We're working really hard to try and control that in New Jersey and doing a lot of research related to it.
My name is Heather Desko. I am a Senior Watershed Protection Specialist with the New Jersey Water Supply Authority, where I manage our aquatic invasive species management projects. The Delaware and Raritan Canal that we're standing on today is one of our actual water supply infrastructure here in New Jersey. And today we are sampling for the invasive Chinese pond mussel. And we are using this technique, which is a relatively new and rapidly evolving technique, to monitor for DNA in a water sample. So we're taking water through a filter that then can get sent back to a lab to be analyzed for the DNA of that species specifically. So just the Chinese pond mussel is what we can look for with that DNA.
The effort to determine the spread of Chinese pond mussel has involved the use of environmental DNA. The eDNA has come back positive for Chinese pond mussel, and so the next step in determining whether the Chinese pond mussel is here is to do a physical survey. And so we have a mussel crew out on the water, scuba and snorkeling, to see if they can find any Chinese pond mussel individuals. The survey crew has found a number of interesting species here that we weren't necessarily expecting. A few state-listed species that they found are the tidewater mucket, triangle floater, and creeper. And so it's really good to know that they're here, because then we can manage for them and help have a better understanding of their distribution. For the mussels that have been found out here, we've been taking some genetic swab samples. Those are preserved in ethanol and transported to the lab at Rutgers, at which point their lab staff will be doing sequencing on them to determine what species we have, and they'll be able to identify them that way.
eDNA, or environmental DNA, is genetic material left behind in the environment from animals, typically from food, waste products, just as they're moving around their environment. And so what we do here is we work to go out and we collect that DNA, whether by concentrating it from water or taking it off of surfaces and then concentrating it onto a filter. And then we take that filter, work to extract the DNA, isolate it, and then we use a series of tests or assays to detect a specific organism by looking for a DNA sequence that is unique to that organism. We can effectively sample a large area at a time, collecting samples several kilometers apart downriver, and then we can bring those samples back, and within about a week or so, we can work to extract and do the qPCR testing of those samples, and then we can essentially see which of those sites lit up, and then we can follow up with, say, divers or physical surveys to confirm whether or not Chinese pond mussel could be found at those sites.
No one worldwide has been able to eradicate this species. It has a life history that makes it very difficult for eradication to happen. We're really the first ones to have a chance to do it. Connection with the New Jersey Conservation Foundation formed during the pandemic, and they contacted me directly asking if we could develop an environmental DNA approach to their efforts to confirm that Chinese pond mussel was in fact eradicated from their ponds on their land. Mussels are difficult to deal with in terms of their environmental DNA surveys. They just have a life history and a genetic architecture that makes it difficult to develop these assays. And so the fact that we've been as successful as we have been is very encouraging, and I think speaks well of what we're capable of doing for the state.
This Chinese pond mussel, if it was to escape this sanctuary and in a flood make it into the Delaware River or Delaware and Raritan Canal, they could simply get everywhere. They have a larval stage that is on the gills of fish. The larvae are called glochidia. They attach to the gills of the fish, and that's probably how they got here on the Asian fish that were being imported. And one of the reasons that makes them so invasive is that when they attach to a native fish, as soon as their larval glochidia attach to that fish's gills, there's a reaction by that fish, and none of the native mussels, none of their glochidia will attach to that fish anymore. So they not only take over the substrate, use up the food supply as adults, but they also wipe out the host fish so that native mussels don't have anybody to use in their life cycle, which is the most damaging thing of all. So it's 2024, and now we have a much more elaborately designed project to treat all the ponds. The chemical has actually been modified specifically for mussels, and there's been some experience with it. And so we think now that hopefully we'll make sure that we finally rid the system of this population of Chinese pond mussel.
It's really important for us to document everything that we're doing at this site from a scientific point of view and from an educational point of view. Ultimately, we want to make sure that this species doesn't spread anywhere out throughout New Jersey or really in the United States or continent. So we're looking here to share what it is, what it looks like, and how folks can identify it. That way, if it is discovered in other places, we can work quickly to eradicate this species.
This partnership is great. Everybody's working together. Everybody understands the severity of the problem, including Jersey DEP, which, that's very important to make sure that everything we do here doesn't cause any unintended consequences to any other natural resources. So it's a big project. We have tremendous cooperation on the part of everybody.